Sure.
Next
Next
slide.
slide. Thank you. I guess so tonight, the agenda, we're going to go through, as Megan said, the clinical need and then talk about the test. But first, I'm going to give you some background on the science behind this test. And I'm happy to take any questions in the middle before we move on. And we will do a brief pause there. If I could have the next slide. So I think, we were in my group as we were des developing this research program, we really started thinking about what if we could predict which children would be affected by autism. And I think, that is a question clinically that's really important is that because we know the earlier we predict, the better the outcome can potentially be for that child. And MAR autoantibodies were or are a potential pathogenic biomarker of autism risk. And these may define a very targetable etiologic pathway in a subpopulation of women. And I think that subpopulation is also an important aspect of this. This is not all of autism, but it's a very, I think, distinct clinical subtype. Next. So just to give you a sort of an overview of what we're talking about, as I said, it's a subtype of autism. It's anywhere between 12 and 26%, and that range is largely based on what population we're looking at. In the higher end of that would be the high-risk moms who've already had a child with autism, down to sort of the earlier testing. And this is all based on my research in my lab that I'm talking about right this particular minute. And so some women develop autoantibodies to their own proteins. I mean, this is well known. And that in these women, these antibodies are going to cross the placenta during pregnancy along with a healthy IgG, where these antibodies can then find their target antigens and bind to them. And on the right, you'll see two pictures of neurons. One in the top is these early developing neurons showing that they are positive for these antibodies. And this is in the developing mouse brain. And down on the bottom, these are adult neurons from animals that were exposed during gestation. And you can see on the left, a healthy neuron with healthy dendritic spines. And on the right, you can see a MAR exposed neuron, which was because you can see very rudimentary spine development. So we're sort of getting some weird stuff on the screen here. And so the MARA has very distinct behavioral subtypes, and we see more severe behaviors in these children, especially if they have, when we start talking about the clinical patterns, more than one pattern that could give them the MARA phenotype. They have more pronounced stereotypic behaviors, as do the animals that are exposed in our animal models. And we see across species, meaning human, non-human primate, mouse, and rat, significant volumetric changes in the brain. Good timing there, Chrissy. Next slide. And so we've identified 8 proteins that are associated with this phenotype. And in the very original early study, you can see we took fetal brain, ran it in a Western blot, probed it with patient and control plasma, and noted that the bands that were showing up, we see the arrows, were more frequent or were only present in the autism samples versus the typically developing and developmental delay without autism. So that's a fetal brain blot. So we went very long story in between there, and we identified each of what those original antibodies were binding to. And it turns out there are 8 different proteins, and it's combinations in the research side, combinations of 2 or more that gave us the MAR autism phenotype. Each of these proteins has a very important role in neuron development, from very early neurogenesis, neuritogenesis, all the way to axon outgrowth, growth cone collapse, development of synapse. Next. And so when we first began this, we really, once we identified what the proteins were, and we started looking for whether single antigens or combinations of 2 or more gave us a very specific autism diagnosis. And we noticed that there were a few patterns that we only saw in the autism population and not in the typically developing control population. And so those are the ones that are boxed in yellow there. It gave us a very high odds ratio, and you'll notice we did not see these in the typically developing samples. We created a very stringent assay, meaning we wanted to be very sure that we were getting the most specificity. So we had a very stringent cutoff, but you can see in the two STIP1 Y box and CRIMP1 STIP1 below, we had a couple of TDs, but they were still significant for autism. Next. So this is a postnatal study, and we went on to check these patterns in a prenatal sample population. So these are second trimester samples we took from the early markers of autism study. We tested those same patterns that we found in the charge sample that I just showed you, and we tested them to see if they were predictive, if they were present during pregnancy, and they, thank goodness, were. The abundance of the patterns, meaning whether CRIMP1 and CRIMP2, which was lower down in charge, is like the number one pattern. In EMA, that might have differed slightly, but the specificity remained. One caveat to this population is these are not validated typically developing controls, they are general population controls. And that just means in the context of this study, they never sought services in the state of California. So we don't have a way of telling if they went on to be diagnosed with autism. But next. So we've assessed and determined that these are very good biomarkers. I'm really interested in, do they have pathologic significance? And for that, we use animal models. We've done passive transfer studies that demonstrated there is pathology associated with the presence of human antibodies in our animal models. But we wanted to create an animal model that was more reminiscent of the clinical picture. And we took the MAR-ASD peptides that we determined what the epitopes were that the clinical samples were binding to on each of these proteins, and we used those to create our animal model. So we immunize them before they're ever bred, and we know that at the time of breeding, there's no inflammation left. There's no inflammation in this model. This is not a MIA model. This is solely based on the transfer of these autoantibodies from the maternal into the fetal space. And we determined that there are behavior changes. As I said, we've got MRI and MRS spectroscopy. We've done immunohistochemistry to look for localization, and we've done cell culture studies to determine if we can tell that we're inhibiting with things like neurite outgrowth in developing early progenitor cells. Next. So we see that these translational models, they display altered social and self-grooming behaviors. Self-grooming is what we can use as a proxy for repetitive behaviors in our clinical sample. By no means do we create an autistic mouse, because that just, that is an artificial label. But what we do see is that we see a reduction in their social behaviors, and we saw an increase in their repetitive behaviors. On the left is the mouse, on the right is the rat. And so it doesn't matter what the genetic background is of these animals. As long as we can break tolerance in the moms to these proteins, we can get the phenotype in their offspring. Next. So we saw decreased communication, decreased social behavior. The rats are incredibly social, and their behaviors were definitely reduced. We saw increased repetitive grooming, and we saw differential expression of the relative volumetric differences, different in the male than the female. Different regions were impacted. The behavior phenotype is not different in the male and female. It is that, so that remains a puzzle as to why, but gives us maybe some insight in how the male and female brains develop differently from each other. I mean, we know that, but this gives us a little bit different context for that. Next. And so, as I said, across all species, including our humans, which are on the right, and our monkeys on the left, that we see this altered brain volume in the ASD exposed animals. That was a passive transfer experiment into the non-human primate. And in the clinical population, the MAR-ASD actually separated significantly from the ASD without MAR and the typically developing population. You can see a much broader range in the ASD non-MAR, and a very tight, nice, elevated total cerebral volume in the MAR ASD exposed kids. Next. So I think, one of the things that we saw in our passive transfer in monkeys is the mothers of the IgG treated was overprotective, which is also going to impact their social development. As they aged, the IgG treated offspring, exposed offspring, showed evidence of abnormal social behaviors with both familiar and unfamiliar pairs, because they're pair raised in familiar dyads. And then the IgG treated animals failed to engage in species typical reciprocal social interactions, meaning whatever they were projecting definitely was not received as a typical social interaction by their peers. Next. So just the takeaways from this very brief overview of the last, 27 years of my research, that gestational immune dysregulation definitely can contribute to the risk of altered neurodevelopment. These maternal autoantibodies to developing brain proteins cross the placenta. We know that. We've tested that. They impair neuronal development. We've shown that. They're similar to the processes we see in hemolytic disease of the fetus or newborn, or congenital heart block in the offspring of systemic lupus patients. Large epidemiologic studies support these patterns that we've first demonstrated and now have proven to be specific. These antibodies localize the brain of the developing animals. We've shown that. They co-localize with their cognate antigen. The mouse and rat models capture all three domains as best we can in a rodent model of the social, communication, and repetitive behavior. And we see the enlarged brains, which I think is a very interesting clue that we are physiologically changing the way the brain is developing. And we're still trying to understand what is causing that. What is that derived from? And we also have seen, interestingly, that gestational exposure to MAR antibodies alters offspring brain cytokine and chemokine expression in our rat models. And this is not in the direction that I think everybody expected, which would be increased, as we would see in a MIA model, maternal immune activation model, with increased inflammation, but it's actually a deficit. And it's a very strong deficit that lasts throughout into adulthood in these animals. So it's a long-lasting impact on what would be a typical profile of cytokine chemokine production by neurons and microglia in the brain. So with that, I will take any questions on the science. And this is my great team that contributed to this work. I am just presenting work that they are working on.
Okay. Yeah, so I'd like to first set kind of the stage for the clinical need. If you go to the next slide, I mentioned in the beginning, I have a background as genetic counselor, specifically in a laboratory that was offering the common genetic tests in postnatal neurodevelopment. And one of the number one questions that a lot of families would ask is, what is the likelihood of having a child with autism? And this, question was also reflected in an independent survey that MarBio did early on to determine, what are the questions that are being asked by families who have a child with autism or a family history. And recurrence risk was definitely a top question, as well as wanting to be able to provide the best care for their child and for them to develop into the, best version of themselves that they can. So, parents want to have this information, not only for the family planning and enabling the earliest possible intervention possible. Next slide. But here's where the challenge comes in, from my experience working in the genetic space is, we know that autism is highly heritable. Many studies, say up to 80% across multiple large scale, family twin studies, et cetera. We see strong familial clustering. There's a high suspicion for genetic influence. Yet, in my experience, even offering, some of the most comprehensive genetic testing, like whole exome or even whole genome sequencing, genetic testing really only identified a pathogenic or, an underlying genetic cause for autism in about 10 to 30% of cases. And so, we see autism occurring, in multiple children across, families. Genetics, or at least an identifiable genetic cause, isn't common in these families. So, what else is going on here? I think this gap really represents a critical opportunity for not only clinical advances, but clinical information for families in navigating this. Next slide. So we want to talk about the MAR autism test. You can go to the next slide. And building on the academic studies that Judy described, we undertook our own clinical validity study to demonstrate the performance of this validated test in a CLIA laboratory environment. So from 907 study samples, so patient samples, we demonstrated that there's a clustering of high-risk autoantibody combinations. If you recall from Judy's slides, there's a number of different combinations that you identify in different populations, some of which have higher risk associated with autism, or what Judy showed, higher odds ratio. And so what we identified was similar, clustering of a very high risk autoantibody combinations. And those combinations that are included in our clinically validated test are located there on the right. These combinations, when identified, result in a 97.5 positive predictive value for this test with a 99.8% specificity. So when we were looking at the clinical offering of this test, obviously there was a lot of background evidence that has been done in the academic setting, but in order to offer this for a clinical setting, we really wanted to minimize the chance of false positives, and in doing so, identifying these highest risk combinations so that there was confidence in the clinical use of this test. In our clinical validity population, the sensitivity or detection rate for these combinations shown here was about 6.2%. Again, because we wanted to optimize for the highest specificity possible. So as far as the validated indications for this testing, it does include mothers who have a child diagnosed with autism, and that can be used to inform upon the recurrence risk in the preconception period. Do you want to note that this is not currently validated for use during pregnancy? So it has to be in that preconception period of use for that recurrence risk information. Or it can be used postnatally to identify and triage at-risk children before potentially even symptoms emerge. The second indication for the test is for families with a child showing signs of autism. So, that's going to be different depending upon who they're presenting to in that kind of clinical care pathway. But, there's a concern for autism. Maybe they're not meeting milestones. Maybe they have shown as high risk on a common screening tool, but they haven't yet received a formal diagnosis. And this could help triage care depending upon, how your different clinical care pathways work. This could help triage care for those patients who may be stuck on long wait lists waiting for that diagnosis, and could help clarify that etiology ahead of time. So next slide. I want to put it into context as far as what this information provides. And again, I'm kind of pulling from my genetic counseling background. So on the left, when we're thinking about a family who has one child with autism, let's say that genetic testing came back non-diagnostic, nothing suspicious, and they say, what is the chance for us to have another child or a future child affected with autism as well? Well, we really just had to pull on kind of general population observations, which in general, plus or minus, a few percentage points depending upon specific family history. But in general, that would average out to be about 20.2% risk that we would give for them to likelihood for, a second child to be affected with autism in their family. Now, if we think about the scenario where a child is maybe showing initial symptoms of autism and they have a positive, let's say an MCHAT or another, screening tool for autism, in general, that's about a 58% likelihood that that child will go on to receive an autism diagnosis, higher for developmental delay diagnosis, but autism specifically, 58%. Now, in these scenarios, if the mother was to receive the test and have a positive result on the MAR autism test, so one or more of those high risk combinations that I showed on the previous slide were detected, that risk now goes up to 97.5% for that family. So 97.5% that they would have a future child affected with autism, or if they have a child that's showing concerning signs right now, or even pre-symptom, early symptomatic, pre-symptomatic, they have a very high likelihood that they would go on to have autism because these maternal autoantibodies were present during gestation. Next slide. So I like to go through just a clinical use scenario to kind of put these different indications for testing and use of this test and information. So, in this case, a 36-year-old mother visits a pediatric neurologist for follow-up, and her 7-year-old daughter had received an official ASD diagnosis. And, while discussing school supports, next step referrals for the daughter, common questions. Mom says, hey, my 16-month-old son is here. Is it too early to learn if he might also have autism as well? Maybe she doesn't have any developmental concerns right now or hasn't noted any significant missed milestones. But that worry is there for that mother, and she wants to know, is there anything she can learn before, concerns emerge? Well, in this case, the MAR autism test can be ordered for the mother. She already has one child who has an autism diagnosis. And in this case, the younger sibling isn't even showing signs, but she does meet the indication for testing because she already has one child who has an autism diagnosis. And let's say if this test came back positive for this mother, it would indicate greater than 97% risk that that 16-month-old would go on to have an autism diagnosis. So, in thinking about that kind of clinical information for that parent, one, kind of the worry that can help, kind of provide an answer, and more importantly, kind of a plan of action for that is what can we do now, get them into an evaluation pathway, and in the meantime, what early intervention services could be provided at that age. So, even when the younger sibling is not yet showing signs of autism, that family can take that information and, act on it. Next slide. So just a little bit of a summary again of the clinical impact. Already talked about, in this previous case example, earlier monitoring before symptoms emerge, triage high likelihood children, depending upon the diagnostic wait list in your various areas of practice. Inform upon potential support need. Now, this is early information, and Judy can speak a little bit more towards the academic studies on this, but preliminary information does indicate that this may be higher support needs for this particular autism subtype. So knowing that, the family can potentially better prepare for what that means for the child's care and interventions. Of course, informing for family planning decisions. Family planning decisions can look, like come in a lot of different, ways for the family, depending upon where they're at. But surrogacy may be an option for them. This is a gestational exposure. Could a surrogate provide, that kind of pregnancy environment that mitigates this maternal autoantibody exposure? And then finally, I see that this a lot, or I saw this a lot in the genetic space, is um, a lot of guilt around the families, the parents around, why did this happen? And, having that information, this isn't anything you did or didn't do. We don't know why these autoantibodies are present to begin with, can kind of help assuage some of that guilt and help families move to, what can we do now with this information to provide the best potential and best care for my child? And I see a lot of questions coming in. I'll pause maybe after this slide and stop and take some questions. So finally, just in the clinical use of this test, I'd like to talk a little bit about where the Smart Autism test fits and kind of the testing paradigm. I know that there's a lot of different tests available out there in families who have a child with autism. This can be used as a reflex following non-diagnostic guideline testing, for example, genetic testing if it comes back non-diagnostic. Alternatively, it could be offered as part of a battery of testing alongside, common genetic testing. And again, I'm talking a lot about genetic testing because that's my background and experience and how I see that fitting into the paradigm. But there were many times when, again, I would talk with families and they've received the most comprehensive genetic testing and they still don't have an answer. And at that time, they said, "What else can we do?" And well, you've done most of the tests that are available. Of course, there may be new discoveries that come along, but this is now a tool that 5 years ago, when I was talking with families, wasn't available to help provide further information about that etiology and recurrence risk. Again, for that kind of early symptomatic child, when a child screens at risk in a common autism screening tool, this test can be done at that time. And then around those families who, want to leave no stone unturned, potentially asking about, likelihood for having another child with autism. So I will pause right there. Let's see. Christy, did you want to go through the questions, or I'm not quite sure where we're at in the list of questions here.
Sure. Look like Dr. Vandewater has answered some of them, but I think some of these questions might be good for the broader audience. So I might just bring them up so we can answer these so the broader group can hear the answer. One of the questions here, Dr. Vandewater, is there any association with the more autism and later development of PANS or PANDAS? Could you comment on that? Looks like you did answer it here in the section, but so everyone can hear, if you don't mind. You're on mute, I think, Dr. Vandewater.
Oh, yes, because my cat decided to show up. The joys of being at home. So that is a study I think that would be really important to do given the immune associated sequelae with PANS and PANDAS. And I think it would be a great study to do. I have not had the opportunity to do that study yet because we've not really had a population, dedicated to that yet that we've studied. So I think it's a great, and which is what I wrote in the comments, I think it's a great next population to look at, actually.
Thank you. Next question here is, why is the test not approved for use during pregnancy? And are there any maternal treatments for maternal autoantibodies?
I can talk briefly about not yet approved for use during pregnancy. We get that question a lot. It has to do with the validation of the test. As Judy mentioned, there has been academic studies looking at blood samples drawn during pregnancy, I think in the second trimester in that EMA study. So we know that it is possible. We just haven't validated specifically for that physiological state at this time in the laboratory environment for this test.
And Judy, anything you want to talk about further for maternal treatments for maternal autoantibodies? Or you want to add to that at all?
In terms of treatments for maternal,
Yeah.
that is something that is actively being studied in my lab using our animal models right now. Actually, we have some trials going for some potential therapeutics. And of course, they have to be safe during pregnancy, which is going to, limit a little bit what we can do. But I think stay tuned for that, because that is definitely something, that we are trying to target.
Thank you. Another question's come in here from Dr. Cohen. Are there any studies looking at kiddos with confirmed MAR autism to see if there are certain types of therapies that seem to best help these children more specifically?
I think that is a very an important area that we are working with the Mind Institute to address because of the clinic situation there. Being able to follow these kids a little longer, of course, getting funding to do that is a bit of a challenge right this minute, but getting a study put together where we can test, because I think, as we're informing clinicians of this outcome, we want to be able to say, but this is what would work best in these kids, to not, both waste time and resources for things that might be less effective in those children.
Judy, can you comment briefly on the association with tendency towards more severe symptoms?
So I think, the what we've seen both in our research studies is just looking at the ADOS total, the severity score, looking at intellectual disability related to the specific patterns that we saw in the margin study. And we have another paper that's in revision right now on that, especially with intellectual disability. There are certain patterns that will go with that. And then I think that's one of the important parts of this, is that a pattern is not just a MAR risk, but it also will tell you potentially whether this child will be more severe or have, higher or less, like higher needs child versus a potentially lower needs child. So I think those we have some clinical data on, and as we are currently testing, we're also gathering information on the families, on their children, so that we can get a better idea of just what the phenotype is, because I think that, again, is very useful of what the needs might be. I can tell you that in cases where they are MAR positive for more than one pattern, say they've got CRIMP1 and CRIMP2 and CRIMP1 and GDA, those kids have always been on the higher end. I mean, they were an ADOS composite score of 10. So, it is a little bit additive, and that is the other thing. It's falls in the precision medicine realm of what is the pattern, how many of these autoantibodies do they have, or all factor into that part of the equation.
Right. I'm just going to ask this next question for you as well, Dr. Vandewater. Are families with autoimmune diseases more at risk? Have you seen any association there?
In the early studies that we did not. However, that is something that I have a very large study going on right now that we call the margin study. And that is one of the key questions. What is the maternal cytokine-chemokine profile during pregnancy? What are her health risks? We've got a huge health question. I mean, because it's
they're answering my question.
Database. It's a Kaiser based study, so we have all the health information on the mother, so that we can address that more specifically to see if there is risk, which of the autoimmune disorders are more likely to give us that risk.
Thank you. Next question, I think, Megan, for you. Can you comment on insurance? And this specific question is around if insurance would help to cover surrogacy for a mother that tests positive for MARA, but if you could comment just in general, that'd be great.
Yeah.
So I think there's some background questions. Is there, if you're not speaking? Thank you.
That's a great question. I don't have enough knowledge and background to know about the insurance coverage of the surrogacy. I mean, it's certainly a reason why surrogacy would be pursued. Whether that would be a covered indication, I don't know. But it's a good question. And I think it's something that we hope, as this The recognition of the MAR autism subtype, the recognition of this biological mechanism becomes more familiar and more recognized. And, in the future in guideline kind of testing, we hope that it does kind of go that route of insurance coverage to help with alternate paths for pregnancy.
Thanks, Megan. I think this question kind of rolls into that as well, in a way. Is the test ever approved for or recommended for a mother who doesn't have a child with autism?
We get that question a lot. So what we initially launched this test is in a higher risk population. That's a common approach to a new test, new clinical test, where we have a better understanding of the risk and the performance of the test in that higher risk population. We are working on expanding it to women who do not have a child with autism. We get that question a lot. And I think that's a great opportunity and great future of where this test is headed. Right now, we don't offer that, mainly because we wouldn't be able to inform the provider or the patient around what that risk for autism would be. When you move into lower risk populations or lower prevalence populations, the risk information that comes from that test does tend to decrease in that lower prevalence population. So it wouldn't be that 97.5% likelihood. But we need to do further validation and understanding of performance in lower prevalence populations to understand what that actual likelihood that we would quote would be in that population.
All right. Another question. How about the population of women who have been diagnosed with autism themselves prior to pregnancy?
Another good question. We kind of right now default to the indications for testing that have been validated, which is having a child with autism or a child who's suspected to have autism based off of potential symptoms. I don't, Judy, have you had any experience in the academic setting about the mother themselves?
I have not, but I haven't always had access to that information in our population.
One of the really exciting things now that this, test is clinically validated, we have the opportunity to do further testing in different kind of clinical populations and access to different clinical information. But we're also trying to collect information on the patients who undergo testing ourselves. And part of that's in the test order form about the, mother's medical history. But we also down the road want to collect additional information from those families, even outcome information about how that child develops and if there's any, particular therapies intervention. There's still a lot that we want to learn about this autism subtype.
These have all been great questions. Thank you, everyone, for entering, and please feel free to continue to ask those questions. Megan, I think you have another slide or two of content that you wanted to go through, and then I believe David wants to ask a couple questions towards the end as well. So I think we'll continue on, but feel free to ask questions as we go through here.
So just a little bit about how to access the testing. It is ordered online via a portal. We will help your practice set that portal up for ordering. You can enter a new patient. It's just a couple of pages of information that we collect about that patient. You can select a draw process. We recognize that some clinics don't have phlebotomy on site available, which is okay. We're happy to drop ship a specimen kit and coordinate a phlebotomy site that is near the patient and coordinate that process for you. And then once the patient consents and pays for the test, we handle that through the online portal on the patient side as well. And the results are available at the lab about 14 days after we receive the specimen at the lab. The results should be ready. The results are uploaded in that portal account, and we notify you as the provider that those results are available. Next slide. This is a just screenshot of the test report. It's just 2 pages. I know it's small, but I blew up here the positive and negative kind of interpretation. and description. We try to keep it pretty straightforward as what this result means. A positive, again, we indicate that 97.5% risk. A negative, we want to be clear about what this test does and does not do. Because this is a subtype of autism, a negative test does not rule out all cases of autism. And, it just shows that there's a low likelihood that there would be children affected by autism due to these specific maternal autoantibodies at this time.
Megan, there was a question that came in around if the MAR autism will show up only in a portion of offspring or any offspring. Can you comment on that?
Yeah, and maybe I'll have Judy add on as well. So, it depends upon when these autoantibodies are present in the mother, and I think that's a question: is when do these arise? But when these autoantibodies are detected in the mother, they do seem to be persistent across many years. Judy, do you want to comment on that?
Yeah, so I think in studies that I've done where we have multiple children in the study, if they were positive at the time of the first child that was diagnosed, all subsequent children were positive.
And are the antibodies in the breast milk, Dr. Vandewater?
So at that point, they're less likely to have an impact on the developing brain because they are the blood-brain barrier is forming, during in the embryonic period and is more permissive during that time, but it starts to become less and less permissive. The antibodies in breast milk are typically IgA. That doesn't mean that they won't have an impact somewhere necessarily, but we don't know what that impact would be. I have tested for those, and IgG is obviously minimal in breast milk, but we do see some IgA antibodies.
Thank you.
Go on to the next slide. I believe this is the last kind of process slide that we have about the test. It is currently a self-pay option. If it's paid in full, $640, or there's a payment plan that's available where the full price of the test would be paid. We do have financial assistance that may be available, and people can go onto the billing page to preliminary, kind of fill out some information about their family and income that can help determine whether they would qualify or not. Okay, so this is the last slide. We have a great team here of customer support, can talk about billing and clinical questions. Myself and the individual on the bottom there are on the clinical team and can talk with families about the test results from an educational standpoint, where we direct any clinical kind of guidance questions back to the ordering provider, but around education about what the test does, what it can and cannot do as far as information. We're happy to answer those questions from families as well as the providers.